Thank you all for coming. I think this is going to be a really exciting talk. As we're kind of mentioning, this seminar series has been hosted by our center, which is like the new Center in Computational and Digital Health Innovation. We're kind of slowly rolling this out, but as a main part of it, we've been having these seminar series, these virtual seminars for the last year, which a lot of you know and have been joining. So far, we've only had Duke speakers at this event, so we're broadening it out now to invite external speakers, and Dr. Finley is going to be our first external speaker, which I think we're really excited about, and I think this will be a fantastic way of kind of showcasing what else is not only like, what you've seen before, computational work being done at Duke, but seeing what else is going on more broadly in the field, and we're glad you're all able to join us. For reference for people who are at Duke, we will, and I'll mention this again at the end, we will still have a networking coffee hour after to come and talk about what you've heard, meet the rest of the community, and have the coffee hour in Wilkinson's lobby afterwards. And then this talk will be available online, and we'll be posting that, and it will be recorded and set for that. So with that, I'll introduce Dr. Finley. So we have our speaker is Dr. Stacey Finley. She is the holder of the Nichole A. and Thuan Q. Pham Professorship and Professor of Biomedical Engineering at the University of Southern California. She received her BS in Chemical Engineering from Florida A&M University and obtained her PhD in Chemical Engineering from Northwestern University. She completed her postdoc training at the Johns Hopkins University in the Department of Biomedical Engineering and joined the faculty at USC in 2013, where she leads the Computational Systems Biology Laboratory. She has a joint appointment in the Department of Chemical Engineering and Materials Science, and she's also a member of the USC Norris Comprehensive Cancer Center. She's a standing member of, as we were just talking about, the MAV Studies section at NIH, and her research has been supported by grants from the NSF, NIH, and American Cancer Society. She has a whole host of awards from the 2016 NSF Faculty Early Career Award. She was a young innovator she was named a young innovator by the Cellular and Molecular Bioengineering Journal. She received the Leah Edelstein Keship Prize from the Society of Mathematical Biology. She's a Junior Research Award from the USC Viterbi School of Engineering, the Hannah Riesler Mentorship Award. She was the 2018 AACR Next Gen Star, 2018 Orange County Engineering Council of Outstanding Young Engineers. She was elected a fellow of the American Institute of Biological and Medical Engineering and a fellow of the Biomedical Engineering Society. So I think we're really lucky to have her here today, and I'm excited to have it turn over and let you hear her talk. Thanks again.
Okay, great. Let me share my screen first. All right, so thank you so much, Dr. Randalls, for the very kind introduction and also for the invitation to give a talk in the seminar series for the Center for Computational and Digital Health Innovation. I'm really excited to tell you about work from my lab where we are using computational modeling to understand the tumor-immune ecosystem and really use this to gain more understanding of how cells are interacting with one another and how that influences tumor growth as a whole. So this is really fitting within an area of the field called mathematical oncology, where the goal is to probe the complexities of cancer using mathematical models. And this is an appropriate approach because we know cancer is very complex, many different cell types that are present, including tumor cells, of course, but also immune cells and stromal cells, as well as different processes that happen on different length scales and time scales. For example, angiogenesis, which is the formation of new blood vessels. And so we want to better understand how is the tumor growing and how do interactions between cells influence tumor growth and also response to treatment. And so the main goal of my research lab is to predict the dynamics, or quite interested in this evolution of what's happening inside the tumors, and particularly dynamics of networks in cancer. And I will talk more about what we mean by networks here in just a moment. And we can use those predictions of the dynamics to help identify how the tumor is vulnerable and then exploit those vulnerabilities to inhibit tumor growth. And we do this using systems biology tools, which means we're studying the whole rather than individual parts. So a whole network of reactions, a whole network of interactions between cells instead of just one cell or one reaction at a time. And my lab is having a lot of expertise in mathematical modeling, and we collaborate with other research groups who have expertise in experimental studies and even clinical applications as well. So when I mentioned predicting the dynamics of networks in cancer, I'll talk to you today about two different sets of networks that we're quite interested in. The first is networks that are happening inside of cells, so intracellular networks, as well as networks that are interactions between cells, so how cells interact with one another. That will be the second part. And then I have a bonus, which is a good portion of the talk as well, to describe how we're calibrating our mathematical models to tumor imaging data. So starting with part one, which is studying intracellular networks and specifically gene regulatory networks, and that is inside of T cells. So we know that given some stimulus, naive T cells will differentiate to become to go to a terminal state, which might be a terminal state that is able to promote tumor death. So we call this the pro-memory proliferative state, or it can go towards a terminally exhausted state, which then permits tumor growth. And so it's important to understand how the gene regulation and the expression of different genes influences which of these states it will go to, because that influences what the T cell is able to do. And so we've developed a body of work that we are using to, first of all, predict the expression of genes in different subsets of T cells. And so I'll talk about that in this first part of the presentation, where we've developed a mathematical model to simulate the gene regulatory network within T cells. We can run that model to look at both population level and single cell level gene expression patterns, and then also use the model to predict the effects of interventions that aim to alter gene expression, specifically to inhibit that exhaustive phenotype of T cells. So this is all work in a paper that we published towards the end of last year, where we have this data-driven mathematical model that predicts the evolution of T cell states. And the kind of model that we're using is called a Boolean model. This is a nice way to represent gene regulatory networks because it allows us to account for the expression of a gene either being on or off, so 0 or 1. And the premise here is that it allows us to account for the switch-like behavior during regulation of T cell functional states. So I'd like to just give you a little bit of an introduction to Boolean modeling, and then we'll go into how we're using it in this particular project. So when we have a Boolean model, this is just a simple example with 3 genes, and we see which genes are present, and then we also have arrows that indicate how the genes influence one another. So in this case, we have a way to update the expression of genes in the next time step based on the rules that govern how they influence one another. So if gene A is going to turn on in the next time step, first of all, it already has to be on, and that's part of its update function. So it has to already be on because it's influencing its own expression. And gene C cannot be on, and we see that with the red arrow with the bar at the end. So this is just one way that we can, first of all, describe how the expression of gene A will change in the next time step. Similarly, we have update functions for gene B and gene C. For gene B, we know that gene A has to be on or gene C has to be on. And then for gene C to turn on in the next time step, only gene B must be on already. And so what we can do is to apply these rules and see how the expression patterns, the gene expression patterns, change over time. Now, again, this is a very simple model, but in the case of T cell differentiation, we have a 25-node gene network where we have different genes that are related to proliferative state, which are shown in pink. We have genes that are related to the exhaustive state, which are shown in green. Then we also have important receptors that mediate the activation of T cells. And so this gene expression network that represents the gene regulatory network was previously published in a paper by Belluri and co-workers. And we took that up and used it to study the evolution of the gene expression network in T cells. So what we can do is to start with this network, and we have the rules written down. There are 23 different rules. We can apply the rules in a random order in order to account for the stochasticity of the cellular environment, and then simulate forward in time to predict what genes are turned on and turned off at any time point. We always start with the same initial expression. The T cells are in this naive state, but by applying the rules in a random order, then we get a whole range of possible behaviors. So one of the main outputs from this model is the fraction of pro-proliferative genes, or PP genes, compared to early exhausted genes, EE genes, that are turned on at every time step. And so I've just color-coded the fraction of pro-proliferative genes that are turned on with pink, and early exhausted genes that are turned on with green. And you can see how the fraction of those genes turned on varies as you move forward in time. Again, we always start with this naive state where there is a greater fraction of early exhausted genes compared to pro-proliferative genes, and then we see how does that ratio, or the number of the fraction of each of the types of genes, turn on over time. So the main takeaway from here is that there are only 3 time steps right in the intermediate points that show a greater fraction of pro-memory genes being turned on compared to early exhausted genes. Overall, there is a predominance of the exhausted genes. And maybe just taking a quick step back, what I've done is to run the model 10,000 times to represent a heterogeneous population of 10,000 cells. They all start with the same initial gene expression pattern, but because of the random order in which the rules are applied, then we have a heterogeneous population of 10,000 cells. And this is just the averaged fraction of PP and EE genes that are turned on at every time point for that population of 10,000 cells. Okay, so when we see that this is happening, then we can also run the model many different times and see, well, what are the, almost like a steady state, what are the attractor states that are reached? And that is a state where we have a certain level of gene expression, certain genes are turned on or turned off, and even continuing to apply the rules does not change that final state that's reached. And so we can look at, well, what are the final states that are reached, these attractor states, and out of the many possible states that could be reached, really there are only 8 attractor states, or 8 end states. And so here I'm showing the frequency distribution of those 8 end states, where you can see we've just numbered them in state 1 through in state 8, just based on the number of those 10,000 cells that fall into that final end state. Okay, and so we see that overall, I've again colored these based on whether that end state corresponds to a proliferative state, that's in pink, compared to an exhausted state, which is shown in green. And so again, we see that the majority of the cells are going to this terminally exhausted state, which also aligns with our experimental observations that show that with consistent activation or stimulation of T cells, they predominantly would go to an exhausted state. Now, what's maybe a little bit more interesting is that out of that 25 node network that I showed previously, each of these end states really only consist of a set of 7 core genes. So just as an example, if we look into end state 1, it has to do with just these 7 core genes, where I have again colored them pink if they correspond to a proliferative state or influence proliferation, or green if they correspond to a state that is influenced by exhaustion. And in this case, we have 2 of those genes that are turned off, and they're color coded as gray. But we have this same core network, just differences in whether the genes are turned on or turned off, depending on which of the 8 end states we're talking about. So what I want to do next is we can look at almost, this 7,000 number of cells that fall into end state 1 and take just a single cell example and say, well, what is its trajectory over time in terms of the gene expression that's happening? And so this is just one example of a single cell within end state 1. And what I'm showing you now is for this example representative cell, we can look at which of the genes are turned on as a function of time. And so I've again color coded the genes. If it's a green box here, it means that that gene is influencing the cell to go towards the exhausted state. If it's pink, it means that that gene is influencing the cell to go towards a proliferative state. And if it's white, that means that that gene is not turned on at that particular time. So this now is really a heat map that tells us which genes are turned on and off at any time point. And so what we can see is that there are some different patterns that arise. For some of the genes, they turn on and they stay on for the remainder of the simulation, even up until that attractor state is reached. Other genes oscillate in terms of their expression, so being off and then on and off and on and so on. We see similar patterns for proliferative genes as well, where some of those genes will turn on and stay on, and some might oscillate on and off. So this allows us to see, well, what are the dynamics of the gene expression patterns that we see inside of individual cells that are falling into this first end state shown here. Now, further, because of the mechanistic nature of this model, we can not only say, well, what genes are turned on and off, but also identify what is the trajectory of the cell state. So it goes from this naive state as a T cell to this terminally exhausted T cell state, but actually there's some oscillations between a pro-memory phenotype as well as the exhausted phenotype. And we can even say, well, which genes are influencing this transition or this oscillation between the two different states. And this is interesting to us because it represents targets that we might be able to go after through collaborations with experimental research groups to say, well, if I want to inhibit the transition from a pro-memory state to an exhausted T cell state, then maybe I should target this gene BCL6 or another gene TCF1. Okay, so we have a very mechanistic model that gives us the expression patterns of genes that we know influence the T cell's terminal differentiation, whether it goes to exhausted state or a pro-memory state, and we can further identify which genes to target in order to inhibit certain transitions, or really to guide the cells into a desired state. Now, this leads really nicely into the next set of simulations that we did, which are to simulate therapeutic interventions. Now, you've probably heard of the checkpoint inhibitor PD-1, and it's a cognate ligand that's expressed on T cells, which expressed on tumor cells, which influences the T cell to go towards an exhausted state. PD-1 is really important in doing this, and there have been interventions that block the PD-1 activation, so immune checkpoint inhibitors, which is a very exciting and active area in cancer research to go after immunotherapeutic strategies. And so PD-1 does appear in our gene regulatory network, and actually in the network, it is actively promoted by a gene called INFAT-C1. And so we thought to inhibit that activation of INFAT-C1, which would then inhibit the activation of PD-1. So we're trying to block this particular edge in our gene regulatory network with the goal of seeing whether it would now allow further T cell differentiation that would go towards a pro-memory state. And ultimately, it's not part of this model, but ultimately, if it's in the pro-memory state, then it would promote tumor cell death. So we're really trying to simulate this immune checkpoint inhibition through a genetic alteration of the gene regulatory network. So we can run our model to look at what happens when we alter that gene regulatory network so that the link between INFAT-C1 and PD-1 is no longer there. And we again run the model 10,000 times to simulate 10,000 single cells. And we run the model to apply the rules in the random order. And now what we see, compared to the wild type, which I showed you before without any alterations, we already had those 8 final end states. Most of those are representing a terminally exhausted T cell state. If we look at the time course, we only saw that 3 intermediate time steps show a greater fraction of pro-memory state compared to the early exhausted genes. Now, in comparison to that, when we implement the PD-1 blockade, you should see from left to right more pink, which means more cells are in a final end state that is characterized as being proliferative or pro-memory. And so we see that drastic difference from left to right. And then also, if we look at the time course, we see now with the PD-1 blockade, we see that now the pink bars are higher than the green bars. So there's a greater fraction of pro-memory cell genes being turned on compared to early exhausted genes. And only 1 intermediate time step shows a greater fraction of exhausted genes being turned on. Okay, so just by implementing this alteration in the gene regulatory network, we see a drastic change in the gene expression that is predicted. So we can not only look at what's happening at the population level with these 10,000 simulated cells, but we can also go in and look at the single cell example. This is what I already showed of a single cell from end state 1 in the wild type without any PD-1 blockade. And then when we add PD-1 blockade, we can look at differences in the single cell behavior. So first of all, just comparing left to right, we now see that when PD-1 is turned on, for the wild type case, it would remain on. But in the case where we have PD-1 blockade, it can oscillate on and off, and that allows the cell to switch out of that exhausted phenotype. We also see some other differences. There's a greater expression of these genes that are related to metabolism, for example, NF-kappa B, mTOR, and others, and also this glycolysis gene. So it cycles on and off between the pro-memory and early exhausted gene. And so it indicates that PD-1 blockade allows for prolonged activation and greater activation of these metabolic genes that are associated with pro-memory state, which also aligns with experimental evidence showing that metabolism is a strong driver of CD8 positive T cell differentiation and also function. And we see some other differences. For example, AP1, which is a pro-memory gene, is oscillating on and off. Again, just seeing differences from left to right when we implement this PD-1 blockade. So one of the main takeaways here is that by altering the gene regulatory network to inhibit PD-1 activation, we can shift the cells, a great majority of the cells, into a proliferative terminal state rather than the terminally exhausted state. So maybe just summarizing this first part of the talk. So I presented a Boolean model of gene regulation that allows us to simulate the gene expression profiles of a heterogeneous population of T cells. We can predict the population level responses, so the distribution of the final end states, as well as the time course, the evolution of those end states, and how we reach that final attractor state. And then when we simulate this intervention that inhibits PD-1, it promotes a shift away from the early exhausted phenotype to a pro-memory, pro-proliferative state. We can also look at how that changes the gene expression patterns within individual cells. Now we could take out more cells and look at their gene expression patterns, and there are really a number of different vignettes and stories that I could tell from here. But I want to move to the next part of the talk because it builds on this model of the intracellular gene regulatory network and talks more about what the consequences of that gene regulatory network are when we're considering interactions between cells within the tumor microenvironment. So here I have to introduce another type of model, which is called an agent-based model. This is where we have autonomous agents, individual cells, whose behavior is defined by certain rules. So we might have a rule that says, oh, a cell can proliferate after a certain amount of time, representing the cell cycle length. We have a rule that says the cell might migrate towards another cell, for example, in response to secretion of a cytokine or some other biochemical diffusible signal. We have rules that say a cell can die. Maybe a tumor cell can die because it's being killed by a T cell, or maybe it can die because it doesn't have sufficient nutrients, so die due to necrosis. And then we also have rules that determine how the cells are using the nutrients in their local microenvironment. For example, to utilize oxygen and glucose and produce lactic acid, and that might influence the health of the cell or its proliferative ability. And then linking back to the first part of the talk, we have rules that influence whether a T cell will go towards an exhausted state or pro-memory state. So we really are having more of a multi-scale model because we're embedding inside of each of the T cell agents the Boolean network model that I described first to determine whether the T cell will go towards an exhausted or pro-memory state. Now, just a little bit more about the agent-based modeling framework. It gives us what cells are there because we have cells that are represented by a point and a radius. It tells us where the cells are located. So this is a spatially resolved model. The cells exert forces on one another. And as I just mentioned, the rules define what a cell can do. Now for the talk, to this part of the talk, I'm going to describe an agent-based model that has 9 distinct cell types. So we have, of course, the cancer cells, the disease cells. We have macrophages that can either be in a naive state or an M1 or an M2 state. So M1, like macrophages, are more anti-tumor, so they aim to inhibit tumor proliferation, whereas M2 macrophages are more pro-tumor. Now, we of course know that macrophages exist along a continuum. It's not just these very distinct buckets or types of cells, but for the purposes of this talk, we're just considering naive or M1 or M2 like macrophages. And then we have a number of different T cell types, so naive, exhausted or pro-memory, again linking back to the first part of the talk, as well as T regulatory cells, which are also pro-tumor, and T helper cells, which aim to inhibit tumor growth. So we can look at the interactions between these distinct cell types, and I'm just representing it here for now as this map that shows how the cells influence one another. So we have cancer cells. Cancer cell proliferation and death is promoted by T cells, whether or not they're in an exhausted or pro-memory state, although hopefully you can see here that the arrow is stronger or thicker for the pro-memory case, so it more strongly inhibits tumor cell death compared to an exhausted T cell. But cancer cells can promote T cell exhaustion due to their expression of the PD-1 ligand. And then we have a differentiation of macrophages into an M1 or an M2-like state that are also influenced by T cells. So lots of cell-cell interactions that we can write down the rules for and then use our model to predict, well, how does that give rise to the population of cells in the tumor and also their spatial organization. We also have macrophages and T cells that further influence cancer cell death, and we have proliferation of the T cells as well. So this is just a small fraction of some of the interactions that we have, but it's meant to represent what we know is happening in biology. And inside of the T cell agents, we have that Boolean model for gene expression that determines how the T cells transition from one state to another. And that incorporation of the Boolean model is being pioneered by a fantastic undergrad and actually master student in the lab, Nichole Tangella. So this is one of the main outputs of the model. First, on the left-hand side, I show a spatial map of what the model, the agent-based model produces. And on the right-hand side, it shows the time course in terms of the number of cancer cells, the number of macrophages, the number of T cells in their different states over time. So just starting here on the left, we can see that what happens with the model simulations is that we start with just a small cluster of cells right in the center of our simulation space. They're all cancer cells. They can promote the recruitment of T cells and macrophages, and then they can proliferate and migrate and so on. And so at the end of a 24-day simulation, this is the spatial output that we get from the model. So you can see where the cancer cells are, where the macrophages and CD4 positive and CD8 positive T cells are. So what we find is that we have this tumor mass that is really surrounded by immune cells. The immune cells are primarily confined to the periphery of the tumor, which again aligns with experimental observations from in vivo mouse studies. Now, we can ask lots of questions using the model, and what we got really interested in is to understand how different properties of T cells, specifically CD8 positive T cells, influence this spatial map, the output of our mathematical agent-based model. So we can say, well, maybe in some cases, CD8 positive T cells are recruited more slowly to the tumor, or maybe in other cases they're recruited more rapidly to the tumor. We can also vary what the probability of the T cells is in killing cancer cells, whether having a low kill rate or a high kill rate. Then we can also compare the effects of the probability of the T cell dying, so a low death rate versus a high death rate. So lots of different outputs or changes that we can make to the model, specifically varying the CD8 positive T cell properties. But maybe I'll just draw your eye to just one combination where we have either slow or fast CD8 positive T cell recruitment with a high kill rate and a low death rate. So this is what the simulation looks like over time. So you can see that when you have slow death rate, there are many more macrophages and fewer CD8 positive T cells compared to fast CD8 positive T cell recruitment. Then yes, there are many more T cells, CD8 positive T cells that are surrounding the tumor. Also from left to right, we see fewer cancer cells. So having faster CD8 positive T cell recruitment not only increases the number of T cells that are there, but now that they're there in the tumor microenvironment, they can promote tumor cell killing. So we have fewer cancer cells on the right-hand side. But we want to link back to, well, what happens when we simulate immunotherapy, and specifically linking back to the first part of the talk where we try to understand the effect of PD-1 blockade. So inside of the CD8 positive T cells, we can embed that Boolean model, which we already have even here. And now for that Boolean network model, we can remove the link between INFAT-C1 and PD-1 so that PD-1 is not activated by the INFAT-C1 gene. And when we do that, we see again a drastic shift in terms of the number of exhausted T cells compared to pro-memory T cells. So what I'm showing you is varying the death probability, the killing probability of CD8 positive T cells compared to base. And you can just see how that might change, even for the wild type case without any PD-1 blockade, how that might change the number of exhausted T cells that the model predicts. So this is just a fraction from 0 to 1 of each of these different types of T cells. And so we see, just focusing in on this case where we have a low death probability and a high killing probability, we have a large fraction of T cells that are in that exhausted state. Now on the right-hand side, when we simulate that PD-1 blockade, then now you see that there are a lot more pro-memory cells, T cells, compared to exhausted T cells. And we see that for other combinations of the parameter values as well. So not only do we have this agent-based model that allows us to see, well, how are cells interacting with one another? How does that lead to tumor cell killing? We can also use the model to look at genetic alterations of T cells and how that influences the final state or the final composition of the T cell population, and go further and say, well, how does that change the propensity of killing of the tumor cells? So that sort of wraps up just this talk about the networks of cell-cell interactions. I do want to spend a little bit of time talking about how we're thinking of calibrating the model to experimental data. So maybe for this second part of the talk, you've noticed that I haven't mentioned at all the kind of cancer that we're trying to represent with this agent-based model. And that is intentional for now, because we don't have an agent-based model that has been calibrated and fitted to experimental data. And really, when we think about agent-based modeling, we first had to take a step back and say, well, what tools do we have to fit such a model to experimental data? So we wanted to really exploit the spatial output that we get from our mathematical model. It's not just what cells are there and how many cells are there, but also where are they located. So this is one of the main outputs from an agent-based model. And when we think about calibrating models to experimental data, usually there are a lot of tools to do that for equation-based models. Now, agent-based models have no equations, right? It's probabilistic, it's stochastic. We have this list of cells that are there. We have a list of rules that define the cell's behavior, but there are no equations. And so for equation-based models, there are very well-established tools for performing parameter estimation and calibrating the model to experimental data. In the case where you don't have equations, there are much fewer resources available to think about how to calibrate such a model. And so I had a fantastic PhD student who started to think about how we might be able to calibrate an agent-based model to experimental data. And it's really leveraging the spatial output of the model. So if you think about it, this output here, I'll bring it back, this spatial output that we get from our agent-based model, it's really just an image. And we might be able to have a similar image that comes from a mouse model of tumor growth, where the cells are identified, not just what cells are there, but what state they're in, whether it's a T regulatory cell or a CD4 helper, CD4 positive helper T cell. And so we have images that we can feed into a tool to calibrate the model. So what Colin started thinking about is using machine learning to perform this, to help us to perform this fitting of the mathematical model. So he developed an approach that uses a neural network. Really, it's called a Siamese neural network that takes in tumor images, again, where the cells are and in what state they're in, and an image from our model simulation, the agent-based model. And really what this neural network is doing is to perform dimensionality reduction. So it's trying to cast these images, which again are multidimensional, many different cells are there, many different types of cells are there, and cast them into this low dimensional space. And really we've just selected 2D, although we tried many more dimensions and it still works, but we can use 2D space because it makes it easy to visualize and also think about the distance between the points that are now represented in this low dimensional space. So really what we're doing is to cast these high dimensional complex images, whether it's experimental or from the model, into a low D 2D space where the distance between the cells, it means something. Where if the 2 points here are far apart, then it means that those images are very dissimilar. They're distant from one another. Whereas the closer these 2 points lie within this 2D space, it means that those original images are similar to one another. So now we can define an objective function that we want to minimize. Really, we want to minimize the distance between the points of the representation of these images in 2D space and use that as our objective function for performing parameter estimation. I'm going to come back to this in just a moment, but maybe let's think first about how we need to process these images in order to perform the representation learning. So what we get either from our mathematical model or from our collaborator who has in vivo tumor images from mouse models of breast cancer growth is a list of what cells are there, what type of cell they are, and where they're located. So we have the original cell locations. What we do is to discretize the image so that we can perform size reduction of the image, because when we put them into our neural network, the images all have to be the same size. In reducing the size, now we have not just where the cells are, but actually this image represents the density of cells. So a darker color here means that there is a higher density of that cell type in the original image, and a lighter color means a lower density of the cells. So this is really all the data or image processing that we do, both for our mathematical model image, the images that come from our mathematical model, and the images that come from the in vivo tumor growth model. So I just want to show maybe one more example of this data processing. This is an image that we get from our agent-based model, where now the pink represents expression of PD-1, the blue are T cells here. So the pink cells are cancer cells, where a darker color represents higher expression of PD-L1. So what we do is perform that discretization, and really what we have are multiple channels of information, right? We have where are the tumor cells, where are the active T cells, where are the suppressed T cells, and also what is the expression of PD-L1. So you can think about this like the red, green, blue channels in any image that you might take, and we have to separate those channels of information and now do the size reduction so that we have a density of information from that channel in each of these images. So now it is the density of PD-L1 expression, or the density of tumor cells and T cells, and so on. Okay, so we perform this data processing of the image, and now we have to use that resulting image in our neural network, cast it into 2D space, and use that for calibrating our mathematical model. Now, how do we do this? We run the model thousands of times with different parameter sets, and we can cast all of those different model simulations along with our experimental data into the 2D space, so the learned space. And we can say, well, maybe we're only interested in simulations that are a certain distance away from our experimental image. So that refines and constrains the parameter range. We're not even considering this point out here because it's just too far away from the image that we're trying to recapitulate. Okay, so then we can say, well, what are the parameter ranges here, and do this in an iterative fashion to minimize the distance between our simulated images and our experimental image. And every time we're updating the parameter values, we're hopefully honing in on the optimal parameter values that give us the closest match to the experimental image. Now, we've done this in a few different just test cases. First is using simulated data, right? So data that comes directly from the mathematical model. We know exactly what parameters we need to produce this model, and we try to recapitulate that output with our mathematical model. We do that using a genetic algorithm where we can run the genetic algorithm many different fitting steps forward in time, and we can have a whole population of parameter values. And so the blue curve just shows the parameter values that give the lowest distance, the smallest distance from the image, so they're the best fit. And then the red just shows the mean here. But so we can see visually that there's a nice match between our simulated image and what we try to capture back with our mathematical model. We can also look at how good of a parameter estimation we're able to attain. So these are the actual parameter values that produce that image on the top. And this is the best fit that we get by using this image-based approach for calibrating our model. So we see that we do really capture those actual parameter values with a small standard deviation. This is just an initial test case to see, is it working properly? And then we can try something that's a little bit more complex and realistic. So this is a biological image from a 2D in vitro experiment from our collaborator, Dr. Kuye Shin, here at USC, where the color indicates the density of dead cells in a 2D in vitro tumor model. So higher density of dead cells is shown in yellow or orange here, and then a lower density is shown in dark blue. And so this is the best fit that we would get from our agent-based model of live and dead cells, just showing the density of those cells. Again, we can see the best fit here as well as the mean value from our parameter sets. And in this case now, we don't know what the parameter values are, right? We're trying to infer the parameter values, but we do think that a good test to show that this is working properly is that we have a small standard deviation for each of the 5 parameters that we try to estimate using this image-based representation learning approach. Okay, so that was with simulated data, another case with 2D in vitro data, and then we said, well, let's go big. Let's try our in vivo mouse model data, where we have these images from our collaborator, Dr. Evanthea Roussos-Torres, here at USC in the Keck School of Medicine, and we can separate out the cancer cells from the image versus macrophages, CD4 positive, CD8 positive, as well as T regulatory cells. So what Evanthea has done is to inject mice with a breast cancer cell line, allow the tumor to grow for a period of time, sacrifice the mouse, extract the tumor, and then slice the tumor and perform multiplex immunofluorescent staining, where she can identify which cells are expressing markers on their cell surface that identify them as macrophages versus CD4 positive T cells versus CD8 positive T cells, and so on. And then we use that image and try to fit it with our mathematical model. Now, you'll notice that in the tumor image, there's a large, blank space right here, which is representing a necrotic core of the tumor. So with this breast cancer model, which is for triple negative breast cancer, the tumors have a lot of necrosis right in the center. So we had to sort of think about how to account for that in our mathematical model. This is just our first approach of trying to fit these data. We do, a reasonable job. There's this growing bubble of necrosis right in the center, which is not physiological in terms of the way that we've implemented it in the model. But at least you sort of get the idea that we have in vivo tumor images that we're trying to match with our mathematical agent-based model. Now, there are also some interesting things that are happening with macrophage density. Maybe they're more localized in this part of the tumor, and that's just not something that we capture just yet with our mathematical model. But I want you to believe that as we continue to refine the model, as well as see whether we're missing any important biological features, that we eventually would be able to capture something like this. So maybe just the next few slides are, well, what could we do with that information? We can look at how the tumor progresses in this learned space. So we can represent the simulated tumor output in the learned 2D space and see, well, as we vary the parameters, once we've calibrated the model, as we vary the parameters to explore different aspects of the tumor microenvironment, how does the tumor evolve in this learned space? We could run our model many different times and see that maybe for some sets of parameter values, we see a very distinct difference in the trajectories that the simulated tumors follow in this learned space. So here I've varied the model parameters, several model parameters over a wide range, and try to see, well, how might they cluster in this learned space? And we come away with two very distinct clusters in the learned space, this blue cluster and the red cluster. Then we can go back and say, well, why and how are they different? Well, they're mainly different because of certain parameter values. So the migration bias of CD8 is a strong differentiator between the red and blue clusters. Another difference is the CTLA4 expression on CD4 positive T cells. So you can begin to see how we might use the mathematical model to understand heterogeneity in tumor growth, and then subsequently understand the effect of different immunotherapeutic strategies as well. So maybe I'll just show one more set of results here, and that is to say, well, if we know that the red tumor and the red cluster and blue cluster of tumor simulations separate out as we saw in the previous slide, then can we see how that correlates with the number of different cell types in each of the final end states of that tumor? So for the red cluster, it has very few cancer cells. So any of those tumor trajectories that were in the red cluster, it actually means that at that final state of the simulation, they have very few cancer cells compared to the blue cluster, which has a lot of cancer cells. And we see that that aligns nicely with, for the case of of the cancer cell, the cluster that has just a few cancer cells, they have low numbers of T regulatory cells, whereas the blue cluster has a lot of T regulatory cells. So we can begin to understand the relationship between the size of the tumor and the composition of the tumor as well. And with the spatial organization of the mathematical model, we can even go in further and say, well, not just are there T regulatory cells there, but are they in close proximity to cancer cells or macrophages and so on. So lots of different questions that we can begin to ask and interrogate using this mathematical model. Let me just summarize this last part of the talk, where we have developed a method to represent tumor images either from in vivo mouse models or from our agent-based mathematical model in low dimensional space. We can then apply that representation learning to perform model calibration and to estimate the agent-based modeling parameters. And we can further use our model to understand the dynamics of tumor growth and even spatial effects that might arise due to different immunotherapeutic options. So I just want to show one last set of images, and this is literally hot off the presses because just last week we obtained these images from our collaborator, where this is a whole lung slice where you can see many different triple negative breast cancer metastases that have gone from the primary site to the lung. And so what we're doing is to zoom in on each of these lung metastases and see what cells are there, again, using the multiplex fluorescence imaging. And that will be our input that we're trying to use to calibrate the model so that we can begin to answer questions that are specific to breast cancer. And specifically, right, we can say, well, what is the spatial organization of the tumor? What role do specific immune subsets play, macrophages versus T cells versus natural killer cells even? And how do cell-cell interactions influence the response to immunotherapy? So this is an exciting collaboration that I have with this researcher at the Keck School of Medicine. Okay, so I described sort of in a sandwich, this agent-based model that allows us to predict the temporal and spatial behavior of the immune ecosystem. We used an embedded Boolean model that also predicts the cell state changes of T cells. And we are using this representation learning approach to calibrate the model to experimental data. And we're really excited about this area of research because it allows us to understand the tumor tissue growth as a whole, at the macroscopic level, as well as intracellularly, how different genetic alterations or immunotherapeutic strategies would influence the tumor behavior. So with that, I'll just thank the members of my lab, especially Nichole and Colin and Gina, whose work I presented today. Fantastic collaborator, Dr. Evanthea Russos-Torres, who we're working with to get the multiplex immunofluorescence images, as well as our funding sources. And I thank all of you for your attention, and I'm happy to answer questions.